Okay guys, let's see if I can use this new iPad app to review with you Cellular Respiration. Now, you know how great my handwriting usually is, so we'll see what we end up with here. But this is for AP Biology. Okay, so if you happen to be in Advanced Bio 2, this could help you right now, but it will have a little bit of detail that you don't quite need. Okay, so remember that any kind of respiration we do, we will always start with glycolysis. And that is the first step, no matter what kind of cellular respiration we are doing. Now, after you do glycolysis, we have one of two ways that we can go. And that depends on if you have oxygen present in the cell or you do not have oxygen present in the cell. Now, if you do have oxygen, we call this aerobic respiration. If you don't have oxygen, we call it anaerobic respiration. Now we'll start here on the right with anaerobic because it's simpler. After glycolysis, a cell doing anaerobic respiration goes straight to fermentation. And that's it. That is the whole of anaerobic respiration. Now if we move back to the left, if the cell does have oxygen present, we will first go into the Krebs cycle, followed then by the electron transport chain. So let's move on to a second slide and take a look at glycolysis. Okay, so in glycolysis, remember the first stage of any cellular respiration, we are going to start with the substrate of glucose. So you eat or whatever, you get that glucose into your system and it gets into a cell. So as it gets into the cytoplasm or cytosol, which is where glycolysis takes place, the glucose is first going to take part in what we call the energy investment stage. We got energy investment. Remember, what that means is that we're going to have to put 2 ATP into this in order to get it to actually happen. So those 2 ATP go in and they form what we call fructose, I'm going to abbreviate a little bit, 1,6-bisphosphate. And that's going to have 2 phosphates on it. Now this makes an unstable molecule. Okay, so as soon as that happens, this fructose 1,6-bisphosphate, which at this point still has 6 carbons, just like the glucose started with, is going to be able to break into 2 molecules of glyceraldehyde 3-phosphate, which we abbreviated G3P. I think if I try to write that out, I'm going to run off my iPad. So we've now got 2 molecules of G3P. The last step of glycolysis is to convert both of those G3Ps into pyruvate. Now here's the good part. When we make these pyruvates for each one, we are going to generate 2 ATP and also an NADH. Remember, that's our electron carrier. We're going to need that guy later when he carries electrons down to the electron transport chain. So, for glycolysis, the net totals are going to be 2 ATP, because remember we made 4, but we spent 2 to start with, and then also 2 NADH. So that's what we've got so far from glycolysis. Now let's move on and stick to anaerobic respiration just for the moment. So during anaerobic respiration, these pyruvates right here are going to move into fermentation. So fermentation, sticking with green for the moment, is simply a way to regenerate, let's get messy, NAD+. So basically, we are going to take our pyruvate from glycolysis, combine it with NADH that we just made, and we're going to be able to convert that into a waste product plus NAD+, which can then be sent back to glycolysis where we started. Now that waste product can be a couple different things. If you have yeast, yeast are going to generate alcohol. And that's what would have happened with our sodas that we made if we let them go long enough. Now, in humans, you're going to get something slightly different. Humans are going to generate lactic acid in their muscles. Why the difference? I'm not real sure. But make sure that you realize that in yeast we're going to see alcohol, and in humans we might see lactic acid. Now the only other thing to throw in right here is that when you do make this alcohol via yeast fermentation, you also give off a CO2, which is why our sodas will be carbonated and why we use yeast when we try to bake things or that sort of deal. Okay, so that takes care of anaerobic respiration. We do glycolysis, we do fermentation, and we can send that NAD plus back so that it can start glycolysis over again. So let's jump back real quick and take a look at what happened in glycolysis. So remember, in glycolysis we started with that 6-carbon glucose, and we broke it down into 2 3-carbon pyruvates. So we got 2 of those. Now, if we are going to do aerobic respiration, we need to move on next to the Krebs cycle. But remember, pyruvate can't really enter the Krebs cycle itself. The pyruvate can be moved to the matrix of the mitochondrion, which is where the Krebs cycle takes place, but we have to do the prep reaction before we can actually send that pyruvate into the Krebs cycle. So I think I'm going to run out of room, so I'm going to open a new slide up and we can watch the fate of this pyruvate as it moves. So pyruvate is going to come in to that mitochondrion, and as soon as it does that, it's going to be converted into acetate. Now pyruvate, remember, like we just said, is a 3-carbon molecule. Acetate is a 2-carbon molecule. So when this happens, we are also going to give off a CO2. And remember, the whole reason we do the Krebs cycle, give or take, is to take the carbons that we got from our sugar and turn them into carbon dioxides. So there's the first one. Now we've got 2 more to go here. But acetate also is unable to enter the actual cyclical portion of the Krebs cycle. So what happens next is we will take acetate and we will convert it into what's called acetyl-CoA. Now, acetyl-CoA is formed when coenzyme A joins with acetate. So now we've got the names combined too, right? Acetate, coenzyme A makes us acetyl-CoA, which still has 2 carbons at this point. But the other thing is that we are going to generate one of our NADH when we do this. So there is another electron carrier. Now, acetyl-CoA is able to enter that cyclic or cyclical portion of the actual Krebs cycle. And the way that it does this first is it joins a molecule called oxaloacetate. And oxaloacetate has 4 carbons. So what we get from the combination of those 2 things we call citric acid, which now has 6 carbons. So we're almost back where we started at the beginning with a 6-carbon molecule. Now the great thing is that from citric acid all the way around this Krebs cycle, we don't have to know the details of the enzymes and intermediates. What you should recognize is that there are actually several steps going on as we move from citric acid and come back to the regeneration of oxaloacetate. And that's what makes it a cycle, right? Whatever we started with, we'll move around and we'll regenerate in the end so that we can do this again for another acetyl-CoA. Now, what we do need to know is that during these several steps that occur as we go from citric acid back to oxaloacetate, we are going to generate a couple things. The first thing to realize is that those two carbon dioxides are now going to show up and be given off. So the other two carbons that we started with up here have now been lost as carbon dioxide as well. Now also in this cyclic portion of the Krebs cycle, you are going to be able to create 3 more NADH. So let's see, I'm going to draw them like this. So we got 3 NADH that come off of there. Okay, as well as 1 FADH2, which was just that other electron carrier that's ultimately doing the same job as NADH. Last but not least, one of the steps that occurs here is also going to form an ATP. So for the second time, we are able to form a little bit of ATP along with those two that we formed in glycolysis. And remember, the ATP production in glycolysis and in the Krebs cycle we called substrate level phosphorylation. Alright, so that is the production of ATP during glycolysis or the Krebs cycle. And we call it that because what we're doing is we're taking a phosphate off of some intermediate that's in here, and so we attach it to an ADP to form ATP. So let's look at the Krebs cycle payoff. Remember, you had 2 ATP and 2 NADH that came from glycolysis. Now, for every glucose that goes into the Krebs cycle, notice that's a glucose. So every 2 pyruvates that go through here, we're going to form 2 ATP for every glucose, and we are going to form 8 NADH for every glucose, and we are going to form 2 of those FADH2s. And that's for each turn of glucose. Now this slide's getting messy, but before we leave, we also need to mention one more thing, and that is that we don't just eat glucose, right? We eat fats and we eat proteins too. So where do those enter this whole deal? Well, lipids or fats are able to enter respiration as acetyl-CoA. So notice, lipids aren't going to give us as much energy as glucose or carbohydrates because they come in later than everything else. Now, proteins can be broken down into amino acids, which hopefully you remember, and amino acids can actually become some of those intermediates that appear here with these little dots I'm making in the Krebs cycle. So again, proteins can also enter in, but later than carbohydrates. So this is why carbohydrates are ultimately a little bit better source of energy for you. Now, let's take a look at our third and final part of our mission, which is the electron transport chain. Okay, so remember, we've got inside of our mitochondrion, we've got a matrix, right, which is the far inner part. And we've got then an intermembrane space. And separating those two things, we have the inner mitochondrial membrane. Now, lodged in that inner mitochondrial membrane, we will find our electron transport chain. So remember, first, we've got 4 cytochrome complexes that we name just by their number. So we've got complex 1, complex 2, complex 3, and complex 4. And all of those are lodged in that inner mitochondrial membrane. So they are integral membrane proteins. Now, once we have formed our NADH and our FADH2 back in glycolysis and the Krebs cycle, those guys are going to carry electrons to the electron transport chain. Now, NADH will drop off its electrons at complex 1, which leads us back to NAD+. FADH2 will drop off its electrons at complex 2, leaving us with FAD. And both of these can then go back to the Krebs cycle or glycolysis and pick up electrons again. Now, after we go through complex 1 or complex 2, the electrons will be passed on to kind of an intermediate called ubiquinone, which we abbreviate as U. So the electrons at this point are going to go on from complex 1 or complex 2 to ubiquinone, who is then able to pass those electrons on to complex 3. Now from this point, it'd be nice if we could say that it just goes on to 4, but it doesn't. We go from complex 3 to, you might remember something called cytochrome C, another accessory cytochrome that plays a role here. The electrons can go from complex 3 to cytochrome C, who is then able to pass them on to complex 4. And then, from complex 4, our electrons are passed to our final electron acceptor, oxygen. Right? And this is going to lead to the formation of water, which is one of our basically byproducts when we do this whole thing. So that's what the electron transport chain does with the electrons. But if you notice, we haven't made any ATP here, right? And that's the main thing that happens during the electron transport chain. So what we still have to take into account that we haven't looked at yet is what we called oxidative phosphorylation. I'm just going to put phospho. All right? And what we have to remember is that as we form our electron transport chain, and as we pass these electrons, hydrogens, or sometimes we just say protons, are going to be pushed across this membrane. So I'm going to write over everything here. Pushed across this membrane. This is active transport. So we're going to push these hydrogens against their gradient out here into the inner membrane space so that we build a gradient. We end up with more hydrogen out here in the inner membrane space than we had inside in the matrix. Now because of that, now that hydrogen wants to move back down its gradient so that it can get back to where there is a lower concentration. And it's going to do that by moving through another integral membrane protein enzyme called ATP synthase. So as all these hydrogens move back across to where they started into the matrix, that movement of that movement of protons is going to allow ATP synthase to make ATP, and a lot of it. Okay, remember, every NADH that goes through this chain is worth 3 ATP, and every FADH2 that goes through this chain is worth 2 ATP. So we're going to make a lot of ATP in this last step of the electron transport chain. So let's add everything up. Remember, this is totally an estimate. Scientists don't know the exact number, really, of ATP that come from this whole process. But if we go back and look, glycolysis made us 2 ATP via substrate level phosphorylation. And the Krebs cycle made us 2 ATP via substrate level phosphorylation. The last part, the electron transport chain, is able to make approximately 34 ATP. And this is all per glucose. So at the end of the day, scientists have suggested that we make about 38 ATP for every turn of aerobic respiration when we have a glucose molecule. Now, remember, this is the whole process. Will you have to sit down and write this for me straight up? No. But if you can do what I just did, you will be capable of really answering any question that might come up in terms of aerobic respiration. So I hope that helps. Good luck.